I don't know how you feel, but I feel like we've forgotten a couple of things. Here's crazy, isn't it? Wow. Okay. Okay. Just gotta stop puckering my mouth off. So yes, I see that's chloroplast. I got that figured out. So that's not our chapter. I also see 10, but damn, that's a lot of paper. So I'm gonna flip through here because I feel certain there's things I also should be doing. Ha. You don't probably know this, but this is my very favorite molecule ever in the whole world. Protein. It's a combination of proteins, but that's ATP Synthase, which we do talk about that with photosynthesis, but that I do need. So I'm going to cut this off. So it's right here on the page where you see 10.3, 10.4. I do need that. And this is talking glucose, and okay, so I feel certain that's glycolysis. So I need that figure. Cut it off. And we do talk about electron transport chains in photosynthesis, but not this particular one. This is also. So I need that page. I'm just going to make sure I need the last 2 pages. I do. So everything that's a figure, pull that. After, wow, that's a lot less paper. So I've got my little dangly 10.1, and all I have are just 2 full pieces of paper in this little part that's 10.3, 10.4. That seems much more reasonable. So saving that for spring, and I'm going to put these pieces in my notebook. So I'm going to stop for a second, fight this piece of hair, and I'm going to cut these just to where they're a little bit smaller. I'll be right back. Okay, no luck with the hair. It must be really crazy because it is so staticky. It's going to attack me the whole time. So we're going to deal. Let's start with the scary one. And I really don't like to put stuff like this in here. But on the other hand, since it's pretty tiny print, I'm going to zoom in out a little bit. Come on, then. Nope. What I want you to do there. This is tricky. Oh, that's not wanting to let me keep it, but that's good enough. I don't want you to think, put my head back, that you're stuck knowing what all of this is. Okay? The other thing that you don't have on here, which I know that's probably like, what else could there possibly be? Well, anytime you have this molecule having it, like this is being phosphorylated, right? Well, an enzyme had to do that. That's what they want you to write right here. So this is a back in the day. This is what we used to have to know. You would have to be able to recognize, not draw, that that's glucose, that gets phosphorylated, and then goes through a little bit of a conformational change from the isomer form of glucose to fructose, and then gets phosphorylated again. Now we've got the molecule that's got 2 phosphate groups, which energetically is not stable, and it splits. And we've already done this. As much as it doesn't look like it, we've actually drawn this. Because we've seen this before. I'm going to go back and show you where it is. You know I've said glyceraldehyde 3-phosphate before, and they're saying that you've got, 2 3-carbon molecules created. Let's keep on going. And then they show you exactly where in the steps of converting glyceraldehyde 3-phosphate to pyruvate that you actually get the electrons that are released, that are picked up by NAD to be reduced to NADH, where you actually have the conformational changes that give you enough energy to create ATP. So don't freak out about this. It is, let's be truthful. They didn't start out with glucose outside of the cell, whatever, but they started off with glucose, right? They showed you that it was phosphorylated. They had another step in here that we left out, where that you went from the glucose to the isomer form that's fructose, but it still just had one phosphate group. I could show you both at the same time. Whoops, just kidding. Right? So they just went ahead and showed this little intermediate step. Then they had the, hello, if you add this other phosphate group, we're going to be in a form where we'll split. No, split. Split would be much more fun. But, so you've got, this is what they're showing you here. That's the name of the molecule with the two phosphates. That now, it's go. You put the other phosphate on, it's go. So that tells us right here, we know this enzyme's name. Once it's a go and it splits, we showed that you have this, we wrote the just easy way of looking at it, this DHAP, but that's what this is. And then glyceraldehyde 3-phosphate, we wrote G3P. And then they showed you everything that we just put an arrow here. We said at some point, you're going to get some electrons that are going to be released, and you're going to generate an ATP twice per glyceraldehyde 3-phosphate before you end up with pyruvate. So as crazy as this looks, honestly it's this, right? So let's go on here and let's conquer this so we don't have to be afraid. And let's add the things that are of interest to us, that matter. It does matter that in the process of making ATP, which you know I told you, you need to picture, because we're going to come up here and put, this is glycolysis, and you need to picture him as just this crotchety old man. I don't know how to spell crotchety. Crotchety. Is that a word? That's my word. Crotchety old man. He's been around forever. He's really good at what he does. And you can just picture him. I can. I hope you can. He's over here. He's like, well, we've been doing this for forever. It works. You know, we don't use those newfangledy things over there, them whatever mitochondria things. We don't need them. We don't need no oxygen. We just do our simple thing of breaking glucose in half. Now, I know it's not fancy and we don't make lots of ATP, but everybody does it. It's just a good process, right? We can hear him saying that. So what he said was we start with glucose. This is now inside the cell, right? And it's in the cytoplasm. We learned that's super duper important because that's our evidence that this process is old. Glucose came in. When we phosphorylated it, we claimed it. Remember us talking about that? So we don't really have to know what this enzyme is, but we do need to know that at first we're going to spend ATP just to get this glucose to where that it's energetically unstable enough to split. So this is often, they don't have the list, they have this down here, but they don't have it up here. Right through here, this part, right through here, is called the energy investment phase. So you spend 2, you make 4, you net 2. Okay, let's go and put what else we know. We know this. Once you add this last phosphate, this is our spot right here, remember? Because this is go. So that means right before it, that must have been the go, no-go part. And this enzyme, which is what this arrow is going to show you, when you go from the molecule that has one phosphate and you add the second one, the enzyme that does that is phosphofructokinase. So we don't have, we know, right? We know it's phosphofructokinase is the enzyme that's going to do this. And that at this point, since we're showing the process here as going, this would be low ATP concentration in the cell. That's the condition why we would go, because this is go as we went this way. And we're going to just go ahead and put, we already said what this is, right? It forms two different isomers of the same molecule. This is our D hap. This is glyceraldehyde 3-phosphate, so it's G3P. And they're saying that they go back and forth and back and forth. Honestly, they both become G3P. So we'll put that in there. And then they're saying, now that you've got these are 3-carbon. All we're doing is just adding on the details that we know, a lot less scary. We're going to come down here where they've chunked this separately, because this is now where you start getting the payoff. So as you move from this unstable phosphorylated molecule and you help it, so if you look through here and you can see still phosphorylated, right? There's two there. There's just one here, one here, one here, none here. Every time we lose a phosphate, it was given to ADP to form ATP. So we can put that on here. We went from, bis means 2, so we went from 2 phosphates, right? We lost a phosphate. Now we're at 1 phosphate, and I always circle them just because it helps. I just did some rearranging, and now I just moved it to the second carbon instead of the third. That's all that means. So I still have 1 phosphate group. I still have 1 phosphate group, and here I have no phosphate groups. Every time I lose a phosphate group, so right here, the phosphate goes to ADP to form ATP. That's where this one that we lost went. Rearranged it. We lost it here. So the phosphate group here, what would I write on that seam, goes to ADP to form ATP. So now we get, just by looking at the molecules, the process involved. It's just a whole lot of just rearranging. This, though, is where we had electrons released. I know that because I formed electron carriers, NADH. So this would be electrons released. And so you would reduce NAD to NADH. And now I've got that to where I've shown we're going to have electrons going to the electron transport chain. So that's not so bad, right? So that just walks us through glycolysis. That was the very first, I didn't say this. In fact, I can go ahead and just show you the order that I put these in. So that was my first little piece that we glued in. Then I'm all about the electron transport chain. I've got these two together, so this is the whole piece with all the complexes. And then this simplified ATP synthase is brought out here. And we'll work with those. And then this is actually like two separate things, all on the same figure. But this became next because these all have to do with other pathways, regulation, type of pieces. And then these look a lot alike, but they're not. Like when you look, you can see here they're saying, oh, wait, you mean we break down proteins and fats as well as glucose? Yes, we do not all eat glucose. That sounds awful. Wouldn't it be an awful life? Ugh. Then this is regulation, and this you don't have yet, but I'll give it to you, and it's just a summary, and then it's going to be where that we build out, which we'll do in class, what goes in, what goes out of every one of these stages. Okay, so where was I? I was here. All right, so let's think about what would we want to make as something we haven't really clearly said. We need to be better about using the words reduced, I gained electrons, oxidized, I lost electrons. So we're going to go through here and we're going to use a highlighter, and it doesn't matter obviously what color you use, but you need two. And I'm going to come over here and I'm going to make myself a little key. So I do like to, up at the top, at least put what this is. So this is the electron transport chain. And I'm going to highlight the, ooh, that's big, highlight the molecules in their reduced form in orange, which means they have electrons that they've gotten, and in the oxidized form in yellow. So this is the color I'm using for the reduced form. This is the color I'm using for the oxidized form. And so to remind myself, they've gained electrons in this form, and in the oxidized form, they lost electrons. Well, if you're handing electrons, like I have them, I gave them, I got them, I gave them, I got them, I gave them. You have a series, that's what we want to be sure we get, that there's a series of reduction and oxidation happening. Like you've got them happening in a row. So let's start off with who has electrons and what we're talking about. So we've been saying, you know, this is the full bus, NADH. It has, see it says, carrying electrons from food. If you have gained electrons, which means I have them on board, you are reduced. So NADH has electrons, it's reduced. We'll talk about this a little bit in class, just because it's, I don't know, higher level stuff and it's interesting to know. But there's a reason why that the parking space for FADH is not at the same parking space as NADH. So FADH2 has electrons as well, right? And what happens, let's just follow one step. So NADH parks in its parking space. This reminds me, like yesterday I went to Kohl's and I had ordered towels and I parked in my parking space and they came out and they gave me my towels. Well, they could have just as easily come out and taken something from me. So NADH parks in its parking spot, and Q takes the electrons from NADH. Now, we haven't talked a whole lot about this, which we will too. Q, because you see it's sitting within this bilayer, it's hydrophobic, and it has the ability to just whoopity-doo go up and down through those phospholipid fatty acid tails. So it literally can move. It's known as a shuttle. So when it gains the electrons, takes them from NADH. Hey, NADH, thanks for dropping those off. No problem, Q. See you in a little bit. I'm going to go pick up some more electrons. Q, I'm just going to do the top of Q. Q becomes reduced. As NADH gave the electrons to Q, it became oxidized, and its oxidized form is NAD+. So we don't have to think about this one anymore. He had them, he gave them to Q, now he doesn't have them. Q does. So Q, as Q can move throughout this bilayer, cytochrome C is not hydrophobic. It's hydrophilic. It's associated with just this outer face of the membrane. So it's going to take the electrons from Q. So thanks Q, no problem cytochrome C. Thanks for taking them. Okay, now I don't have them anymore. So Q has two forms. It has an oxidized form, just like we had with NAD, and it has a reduced form, just like we had with NADH. They have the ability to gain and lose electrons. C up here now is going to, we don't have this here, let's put it, well there's oxygen right there. C is going to give the electrons to oxygen. Oxygen is going to gain the electrons. So when oxygen takes the electrons, it becomes water. Okay? So this is the reduced form. When it did not have the electrons yet, it was in its oxidized form. So when oxygen took the electrons from C, it became water. C was oxidized. It lost its electrons. So having this to where we draw it helps. FADH2 parks at this peripheral protein, complex 2, and Q in its little shuttle form can also take electrons from FADH2. When FADH2 loses its electrons to Q, Q is reduced, FADH2 is oxidized to, I can't read that, FAD, and that's its oxidized form. So all of this that we did, we're showing these different forms. I do better like this. I appreciate that. You know, I'm a very visual learner. I don't know why, but that's messy to my brain. So I'm going to work on this with you in my head. I'm going to have NADH. I don't care where it parks. I can come up here and reference it. But it has electrons on board. Okay? So I'm going to show that it gives its electrons to Q. Q gained electrons, okay? Gained the electrons. Now we'll keep going with this. Just hang in there with me. Now it's got its electrons. It's going to give its electrons to C. We're showing C right here doesn't have any. It's going to get them. Now it has them. C is going to give its electrons to oxygen. Oxygen plus electrons equals water. Okay, let's see how that goes. So NADH in this form has electrons, right? It gives its electrons to Q, which means that NADH forms NAD+. That is the oxidized form. So this is in its reduced. I've got electrons. I just gave them away. Now I'm in my oxidized form. Q, as it was before, originally had no electrons, gained them, and became reduced. Gave its electrons to C, which had no electrons, gained them, and became reduced. Gave its electrons to oxygen, which wasn't carrying any, gained them, and became water. And we can do the same thing here with this. We could put FADH right here, right? 2, because it's also going to give them to Q. And we could put FAD here. I had electrons, Q didn't. They gained them, now Q does. Q gave them, C didn't have them, now he does. He gives his electrons to oxygen, who didn't have them, now he does. I don't know if that made that better or if that made that worse. But I hope that what we get is the pattern of, what did I make you orange for, crazy woman? I hope we get the pattern of this series of redox. That's reduction, oxidation. I had them, I don't have them. I have them, I don't have them. Them being electrons. Okay? Alright. I'm sorry if that made that worse. If it did, just forget it. I hope it helped. Like, I have to see a pattern. And this is, blah. It's beautiful. It's one of my favorite things in the whole world. Because of just the spectacular beauty of it. But, okay. Pitiful. Pitiful. Let's talk about this molecule. Oh my goodness. Come on, P. Yes. If there ever was something that should wear a crown, ATP synthase should. So back in the day, like even before me, so pre-dinosaur times, we knew what this molecule did, and we imagined how it might do it, but you think you're crazy because what you're describing is this equivalent of like a water wheel that's going to spin. Oh, wait a minute, you're crazy. Isn't ATP synthase like an enzyme complex? Yes, I know, but based on evidence of what I'm observing in the lab, I think that's what it would have to do. So it's very much like one of those revolving doors at a fancy hotel or at a fancy like department store, and you know where some really like 12 of you shouldn't go in one spot, so everyone goes in its own little spot, and as it moves, it's going to take you out on the other side. That's what this is. So let's remind ourselves, okay, this is like the closet. Remember? So if we drew our stuff, we would draw that this is that dark yellow with all the just hydrogen ions in here, like a closet, right? So this is the intermembrane space. I know it says it there, but I'm going to be truthful. I don't see stuff like that. I don't see it. I see this. My brain sees this. I swore I wasn't going to ever write this again. Okay, that's the intermembrane space. It serves no purpose. It is a closet. What's it a closet for? Hydrogen ions. So we'll make sure we put some to know. That means in here, this is the matrix. This whole thing here is what mitochondria have. So they've got these two locations. Let's see if I can do this just. I don't know. That's good enough to show that we don't have very many. So we have a gradient, right, of hydrogen ions. Lots of hydrogen ions in this inner membrane space storage part. Not very many in the matrix. There's a natural force. Besides the fact there's a lot of forces, let's be truthful. If you put a bunch of positives together in a small space, they're going to repel each other, right? There's this electrochemical force. There's this motive force of entropy. This has, entropy's disorder, right? Everything moves to disorder, which is randomness. This isn't random, obviously purposeful. If I come in and your sock drawer is all KonMari'd, and if you don't know what that is, you can go watch Netflix and watch her, and you've got them all rolled up and they're by color, and I know you did that. That didn't naturally happen. This wouldn't naturally happen. So it has low entropy, low disorder. Natural things move to high disorder, random. So it's going to naturally move from, this is a high concentration of hydrogen ions. This is a low. There's going to be a natural movement that it would go this way. Well, we can't, because this membrane right here, this is the membrane. This membrane is not permeable to charged things. So what could I do to make this look more like a membrane? Did that help? Okay, so this is that. Why am I yellow? How can I miss that? Here. It's this inner membrane that we've seen that's all folded. Okay, all of that being said, this is your ticket out. This is ATP synthase. Let's look at what it has. It literally rotates. So the hydrogen ions have this little input that they fall into. You know they're gonna, because there's gobs of them. So they're going to go through here. They're going to attach themselves to these little domains, these separate parts of this ATP synthase. This is the rotor part. And this is this, what they call this the stator. They're saying this is kind of what anchors it. So this is the anchor. So this is what spins. So cool. And as it spins, after so many turns, you're going to have hydrogen ions come out. So let's make sure that we understand that this little guy went on a trip around, right? Came out here on the other side of this inner membrane. That mechanical energy causes this particular part, it's called the catalytic knob. But what kind of happens is, if you hold one thing stationary and you crank another one, think about the force you get. And then if you not really let go, you could do that, and that would release energy in motion. It wouldn't do anything. But if you cranked it, and you left one part stationary and the other part cranking, you build up this mechanical tension as energy, and then it's used to create ATP. So ATP synthase is going to use, mechanical energy of this rotor, and it's going to transfer that energy is going to be transformed transfer that energy through the catalytic knob. Catalytic. So catalytic would be like, I don't know if you've ever heard of things in your car, like catalytic converter, do you know what I'm talking about? Anyway, it's saying it's the place where things happen. Like that's where that we're going to convert the energy from ATP, and we'll use just inorganic phosphate that's also in the matrix to form ATP. So the way I wanted to do this, and the reason why I had this page, is there's words that they're going to use that you need to picture in your head. Okay, that's weird, but I know what that means. So here are some terms that are going to be associated with this process of using a gradient with this particular mechanical energy to chemical energy converter, that kind of thing. So one of them is called proton. Okay, we know those are hydrogen ions. Motive, movement, force. That's the natural tendency of them to move with their gradient. That's what they'll use the words for. That just tells you that you have a hydrogen ion gradient. Okay, that's one thing. The other thing that they could call this, and we talk about it all the time, is oxidative, I'm scared of this computer, phosphorylation. That is the process of using a hydrogen ion gradient, that proton motive force, to create ATP, and we have to be specific here, using oxygen as the final electron acceptor. It's on the other side. Okay? So that's another way they could say this. They also can talk about, we haven't talked about this, but we talk about this a lot when we do the nervous system, membrane potential. That just means that you have potential energy because you have different charges across a membrane. So this is different charge across a membrane. Okay, so I'm going to blow up. Okay, so those are our fancy words. And let's go back to this one. What does this mean? Well, I have more positives here than here. That's a difference in charge. Okay? That's this picture. I think I'm going to stop this one and do another one, because this is regulation and it's different. And besides the fact you've told me, and I've already gone over a little bit, but I'm doing better. I could have gone longer. Okay, so we'll stop there and we'll come back to another one.